Welcome to Veterinary Physiology Lectures. It is me, Simpson Soren, Assistant Professor in Veterinary Physiology. So today we will learn the genetic control of gonadal development. How the genetic it control the gonadal development. So let us begin. So there are three things we have to learn in this lecture. First, the genotype, SRY gene, and bipotential gonad. So what are the three important things for the development of gonad? It is essential. All three things are essential. And what do you mean by this genotype, SRY gene, and bipotential gonad? So genotype, let us discuss first the genotype. We know that sex determination takes place at the time of fertilization. So fertilization, the oocyte, it will be waiting in the oviduct for the sperm for fertilization. So oocyte, it carries the X chromosome. Here I have shown here two oocyte or the female germ cell. So at the time of fertilization, if the sperm carries the Y chromosome and that sperm take part in the fertilization or it penetrate the oocyte, and as a result of fertilization, this organism will be XY chromosome and that will be a male. This is the genotype. And if the spermatozoa it carries the X chromosome, and when it is fertilized the Y X chromosome, then it forms the XX chromosome, then it will be a female. So this is genotype. The sex determination it decides or the takes place at the time of fertilization. But here it is not enough. The male which carries the XY chromosome, this Y chromosome carries a special information, or the segment of Y chromosome carries the special set of genes, that is SRY, and that SRY have to express properly at proper time. And what do you mean by SRY? This is nothing but sex determining region in Y chromosome. So that is very important for the development of sex code in the testis. If it fails, then the sex code in the testis will not form. But in case of female, it is having the XX chromosome. It doesn't have the Y chromosome, so they don't have the SRY factor. That doesn't require this special expression of SRY factor. So it looks like female is like an inbuilt system. So what is sex determining region in Y chromosome? So it have to express or the onset of SRY expression in the pre-Sertoli cell. These are the Sertoli cell which is not differentiated. So we call it here pre-Sertoli cell. And this onset of SRY expression have to occur in the Sertoli cell. And when the onset of SRY expression occur in the Sertoli cell, it trigger the Sertoli cell. It trigger itself the Sertoli cell for the expression of SOX9. And when the SOX9 is expressed, or this gene SOX9 is expressed under the onset of SRY expression in the Sertoli cell, and it can trigger by themselves SOX9. Once it is expressed in the Sertoli cell, it can trigger by themselves and increase the SOX level. And also SOX9 stimulate other pre-Sertoli cell for the expression of SOX9. And this SOX9 also trigger the Sertoli cell for the expression of this growth factor FGF9 and prostaglandins. So this growth factor further trigger the SOX9 production. Ultimately, all the system they increase the level of SOX9, and that is very important. So that the Sertoli cell will differentiate and form the Sertoli cell from pre-Sertoli cell and differentiate to Sertoli cell, and that is initiated by the onset of SRY expression, and that is carried by the Y chromosome. If it fail to onset the SRY factor at proper time, then SOX9 will not be expressed. Then sex cord or the testis, the main seminiferous tubule, may not be developed properly. So that is very important, which is carried by the Y chromosome. That is why sex determining region in Y chromosome is important. So let us talk about the bipotential gonad. We have learned this sequence to form the sex cord in the testis. And also we know that primordial germ cell, it is developed extra embryonically. And when the primordial germ cell, it migrated to the genital ridge. Genital ridge is the primitive gonad. So at this time, genital ridge, they have not yet decided they are going to be an ovary or the testis, irrespective of their genotype. It may be XX or it may be XY. So the genital ridge you can convert into testis or ovary irrespective of their genotype. So here, when the primordial germ cell migrate to the genital ridge, and this primordial germ cell, if the organism is XY male, the genotype is XY, then this primordial germ cell interact with the Sertoli cell. This is which is expressed SOX9, and then they develop the sex cord that in future it will become a seminiferous tubule and the sperm production system. This SOX Sertoli cell surround the germinal primordial germ cell and then form the seminiferous tubule. So that is SOX9 if the organism is XY, and if the organism is XX or the female, then this SOX9 will not be expressed. So automatically this cell become granulosa cell and it will form the ovary. So in the female, because the genotype is XX, they form the ovary. They don't need for a special expression of SRY or SOX9. To become a Sertoli cell, the Sertoli cell must express the SOX9. Then only the testis will come or the seminiferous tubule. But when the female individual, it has only XX chromosome, so they don't express the SOX9, so it will automatically form the ovary, the female gonad. So even though the genotype is XX or XY, especially the XY, the Y chromosome carries the SRY, it have to express to form the testis or the seminiferous tubule. If it doesn't express the gonad, automatically it will form the ovary. So that is the genetic genotype and the SRY factor important, especially for the male. And the female, if there is no SRY, the genital ridge, automatically it will form the ovary. So this is about the genetic control of gonadal development. So thank you for watching my videos. If you have any queries or doubt, please mail me simpson.soren@aau.sa.in.